You guys want to have a crack at these if you can read the small font? Can you read them in the back or should I read them out loud?
Knuckle?
Let's just do one at a time now. I'll pop up an answer for you if you guys want to take notice. What do you think for number one?
For
You
knuckle?
think a carbuncle, not a furuncle?
The furuncle.
Okay, why? What's the difference between those again?
Yeah, one was a bunch of T cells.
Okay. So if it's just a singular instead of a cluster of those things, it would be a what? Furuncle. Yeah. Carbuncle is a little bit bigger of a deal.
So it's a furuncle. So it's D. So it's D?
Yeah. 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 For cellulitis and erysipelas, why wouldn't it be consistent with that, do you think? Why wouldn't it be erysipelas or cellulitis? They're deeper skin infections, yeah. And then what are petechia? We haven't really talked about that yet. They're they're basically at the surface. You get these little. It looks like little rashes, and sometimes they present as rashes. But they're broken capillaries at the surface. They look like they're usually really small, less than 4 millimeters. Those are what petechia are. We'll talk more about petechia. Okay, this next question, I'll just read for you guys in the back. This will be an example of an exam type question. We haven't covered many organisms yet, but we will. What do you guys think? If we're talking about erysipelas, we really only talked about a handful, so you can just eliminate the ones we haven't talked about yet. And then, okay, I hear somebody say Streptococcus pyogenes. Anybody agree with that? Yeah. Okay, I would agree with that as well. Strep pyogenes. Some of these are not mutually exclusive, like they can be caused by both Staph aureus and Streptococcus pyogenes, but when you're talking about erysipelas, that's predominantly going to be Strep pyogenes. Or what Lansfield group? What else do we call Strep pyogenes? Group A strep. Yeah, good. Okay, and then we have sort of like a clinical vignette style question, just sort of desensitizing you guys to these so you don't get freaked out on the exam. So we've got an older gentleman who has a comorbidity, he has diabetes, and he's got a painful red swollen area on his lower leg. It's warm to the touch and it's poorly demarcated. He has a fever. Cellulitis, okay. Why not erysipelas? Because it sounds like a pretty prominent. Yeah, so the thing that kind of gave it away more than anything was the poorly demarcated affected area. If it's well demarcated, if it has really visible margins, then you'd be more likely to be erysipelas. And which of those is generally considered to be more severe and a little bit deeper? Cellulitis. Yep, good. All right. So E, D, E. Oh, I'm sorry. Number one is not E. That's Miss Key, bad instructor. It is furuncle.
Points back.
Yeah.
Points back.
Horrible. All right. Next battery of questions. We've got another question about strep pyogenes, virulence factors. Rapid spread of infection by breaking down hyaluronic acid. Yes, we'll never give you something this easy on a test. I'm sorry, I can't do it. You should be offended if I were to give you something that easy.
I was so offended.
All right, question 5. We've got some sort of hemolysis. We've got streptolysin S, virulence factor produced by group A strep. How does it primarily damage cells? Just look at this list and kind of start ruling out things that do not track. And then what do you think is most likely? said it, Sylvester? Oh, I was nodding encouragingly. What was that other license that is produced by Streptococcus? You guys remember? Oh, right. Which one is oxygen labile and which one's oxygen stable? S is stable, easy to remember, right? So the other one is oxygen intolerant, it's not active under oxygenated conditions. So this one is the oxygen stable one, and this is it stays mostly associated with that cell wall. So if you remember that picture, some of it's secreted out, but a lot of it stays there. Whereas streptolysin O, the reason why it's such a good target for testing antibodies is because it's a normal exotoxin, so it's secreted out, and it's very immunogenic. Not this guy, not quite as immunogenic. So what do you think? Any idea at all? Your B, yeah. Did you get that because you knew it or by process of elimination? Did process of elimination get you guys anywhere on that one? Just kind of ruling out the ridiculous ones?
They all have this.
Okay.
Yes.
All right. Yes. Well, it doesn't need to disrupt cell membranes much in the way that streptolysin O does. It's the same mechanism of action, different specificities, but they both disrupt cell membranes and they get rid of a lot of cells, just indiscriminately integrating into those cell membranes and causing them to release. Okay, we've got which Staph aureus virulence factor is a pore-forming toxin associated with severe necrotizing pneumonia, particularly in community-acquired MRSA infections. All right, sounds like some confident people out there. Anton Valentine, leukocyte? You were correct. Let's see if I keep this right this time. Yes. Okay, good. All right, and then last battery of 3 questions for you. Which of the following is the most common mechanism of Staph aureus food poisoning? Why is it not A? Yeah. Yeah. So most of it, you have the bacteria in the food source in that cream puff or that ham salad or whatever that it likes so much. The bacteria are replicating, they're forming that preformed exotoxin, the enterotoxins. You can have a bunch of different types, A, B, C, D. You're getting an accumulation of those exotoxins in that food stuff, and then you can kill the bacteria themselves with those preformed toxins since they're relatively heat stable and acid tolerant. They can survive ingestion, they can survive a trip down through your stomach into your gut where they cause those symptoms of food poisoning. So B is the correct answer, ingestion of preformed enterotoxin and contaminated food. Similarly, if we had to diagnose, just have a crack at this. We talked about this a little bit, but we just talked about there may not be a viable bacteria left, right? So you can eliminate a couple options and maybe rule them out and then arrive at which cancer do you think? Yeah, I'm hearing a lot right. So C, it's a toxin, right? So you can test it with antibodies and see if it binds. And we do that for Clostridium difficile, we do that for some E. coli toxins. So. And then last question, which of the following characteristics best describes the enterotoxin produced by Staph aureus that causes food poisoning? We've also touched upon this. I hear some people saying C. Any issues with C? Everybody understand why it would need to be able to survive the low pH in the stomach. It would need to be able to be somewhat heat stable, probably. Yeah. Okay.
I might have a question.
Yeah.
You could also do letter A, right? Just not.
It's not.
Bacteria not survive.
That's sort of the gist on why that. I mean, if you are able to grow Staph aureus from stool, great. But what happens if you can't? And more than likely you probably can't. It doesn't. There are some Staph aureus in the GI tract, but they don't do very well. They're not one of the more represented species there. So if you got a negative stool culture, even though culture is really a sensitive technique, it would, it could just be that the bacteria died, and the toxins themselves you would expect to find in the stool. So that would be a far more reliable method of determining whether or not it was Staph aureus related enterotoxin release. Okay, so we will, without further ado, start talking about gram-negatives for the first time. We've been talking solely about gram-positives, Staphs and Streps and Mineralcocci. But today, we will talk about some of these enteric bacilli, gram-negative rods. Most of the genera on this slide that we're going to be talking about today and tomorrow in this deck are, they belong to this big family. It's a one taxa above genus. So it's a family called Enterobacteriaceae. They share a lot of common characteristics. Most of them are heavily colonizing of the gut, the human gut in different places, but some of them can occupy different niches as well, like the nasopharyngeal cavity, some of them. So these are all Enterobacteriaceae. Campylobacter is in a different family. It's not a member of the Enterobacteriaceae family. Vibrio, Yersinia, and Helicobacter. So outside of that family. In terms of relevance, if you visit the CDC's enteric pathogens website, their homepage, you can kind of see where some of these members of the Enterobacteriaceae stack on their list of most relevant pathogens and diseases, which they have co-mixed together, which drives me crazy. But a lot of these are going to be members of the Enterobacteriaceae. We have one that we've already talked about, staphylococcal food poisoning, serious food-borne illness you can get, bacterial food-borne illness. But among the members of the Enterobacteriaceae, we have Shigella, we have E. coli, we have Salmonella. Shigella is not on here, but it could be. Typhoid fever, which is caused by certain types of Salmonella, so it's kind of redundant there. And Yersinia. Vibrios, Campylobacter, we'll talk about, they're also gram-negatives, but they don't belong to the Enterobacteriaceae. And then we have a couple gram-positive rods, like Clostridium perfringens, and botulism, which is another toxin-borne food illness caused by Clostridium botulinum. And Listeria, that's another gram-positive rod. In terms of common characteristics, so these gram-negative rods, this is sort of like a mixed gram stain, so you can very easily see some gram-positive cocci. Looks like maybe Staph aureus would be my guess. So some Staph aureus commingling with some gram-negative rods. A lot of these different bacteria are going to be commensals in the guts of humans and animals, and that's important for us to be able to kind of trace and track where the sources of these different enteric bacteria are. Because they're gram-negatives, they have that outer membrane, which helps them be a little bit more resistant to that trip down through the GI tract. They have to survive stomach acids, etc. So they do tend to be relatively hardy. They're also facultative anaerobes, which means what? In terms of their ability, their relationship with oxygen. The Enterobacteriaceae can live in the presence or absence of oxygen. They don't happen to use oxygen as a terminal electron receptor in the electron transport chain, but so they're oxidase negative, which is another test we can do in lab for Enterobacteriaceae. But they can still survive in the presence of oxygen, which is important. And they also ferment glucose properly. In terms of where we find these, there's some distinctions that are helpful to make before we start talking about them individually. Bacteria like E. coli, these are absolutely bread and butter normal flora of our GI tract. They're not the most heavily abundant or well-represented species in the GI tract, but we do have a lot of E. coli in our gut. And most of it, the vast majority, is completely benign, non-pathogenic, just doing its thing, contributing to our metabolism, etc. Klebsiella, this is a genus that has a couple important members. Pneumoniae, as the name suggests, that species name, causes more than just diarrheal disease. It can cause pneumonia as well. So it's a normal colonizer of our gut, but it can also colonize the nasopharynx as well and cause, under the right circumstances, it can cause pneumonia and respiratory infections as well. There's another species called Klebsiella oxytoca, which hardly ever colonizes the respiratory tract, and so it's almost strictly associated with diarrheal infections and not respiratory infections. But it serves as a good example that just because it's a gut bacteria doesn't mean that it can't cause infections outside of the intestinal tract. This guy. Proteus mirabilis, it's another normal resident of the gut. However, this one is associated heavily with a specific type of infections. Anyone ever heard of it or know what kind of infections outside of the intestines? UTIs. UTIs, Proteus mirabilis. So what happens is it's a gut bacteria that gets outside of you and it starts contaminating the lower part of the urinary tract and ascends and causes UTIs. That's a common source of UTI infections. In this middle category, we have some other organisms that are maintained in animal reservoirs. So this has, a lot of these are linked to foodborne outbreaks because these are food-producing animals like chickens and cows, and maybe not so much rodents, but at least with chickens and cows, chickens are heavily colonized with salmonella. So whether we're eating chicken or we're eating eggs, you can count on there being heavy contamination with salmonella. And that's why we really have to wash our hands when we're handling chicken, especially raw chicken. And we have to make sure that chicken is cooked, right, to 165 degrees Fahrenheit because of things like salmonella. O157H7, this is a really extremely pathogenic strain of E. coli, and it is maintained in cattle. So this too, we tend to get big outbreaks of E. coli O157H7 when we have contaminated beef, and sometimes even with crops, because this can run off into water, wastewater from that's contaminated with like cows, and that can contaminate our water with this bacteria. With Yersinia pestis, we've talked about this a little bit in the past. What does this cause? Plague. Yeah, so this is maintained in rodent populations. It affects rodents, and it's transmitted to us usually by what? Fleas. Yeah, the fleas that live on the rodents. Yeah. So most of these animal reservoirs, that flea, by the way, would be a mechanical or biological vector? Biological vector, yeah. It's biting, and that bacteria has to live out part of its life cycle in that flea. So it meets the criteria of being a biological vector. And these bacteria, by the way, when they are in these reservoir species, they do not cause disease in this species. So it's not going to sicken the chicken. It could be normal health chickens, and same with those cows. It's amazing, the cows do not get sick from carrying around O157H7. And then lastly, on this far right column, we have examples of what you would call strictly human pathogens. These are not maintained in animal reservoirs. So both of these are fairly virulent. They have low ID50s, and they can be transmitted from person to person. So poor hygiene is usually what we associate with these, or they can be transmitted by things like flies. I mentioned on that last slide E. coli, and the fact that E. coli is carried in the GI tracts of cattle, right? So when cattle are crapping all over the fields, and that's like draining off into our water supply, that's a big contributing factor for stuff like polluted, contaminated rivers and lakes. You guys ever heard of the term coliform before? Okay. So sometimes when you go swimming, you might see a sign posted that says high coliform count warning. And that is telling you that they have detected high levels of enteric bacteria in that water. The EPA and other agencies set regulatory guidelines for how much we can tolerate of these different organisms in water and what purpose that water is serving. If you're swimming in it, you can tolerate fairly high coliform counts, under 200 of those enteric bacteria per 100 mils is what we can consider the limits for safety. If it's drinking water, we really can't tolerate much. We need to have most of those enteric bacteria out of our drinking water. So the limits there are under 1 coliform per 100 mils. I was just reading the SAWS report, the San Antonio Water Systems report. It literally came a couple days ago. And I was skipped right over all the radioactive stuff and the heavy metals, went right down to the coliform numbers for my particular area, my part of San Antonio. And And sure enough, it was right at that max. They had that one E. coli that they detected in routine surveillance. Yes, sir.
Do you think like one pool, like in the image, if it was like the most pure water ever?
Yeah.
Do you think one turd would tip the scales?
Well, it depends. It depends. Like, that's such a good question. I mean, how big is the water? Is it chlorinated? Is it a swimming pool, or is it like an outdoor lake? If it's an outdoor lake, keep in mind there's going to be a lot of animal waste contributing to it in the first place, right? It's always at your own risk when you're talking about jumping into a lake. But like in this picture, this is a swimming pool. Anybody know what movie this is from? Caddyshack. Good job. Yeah. Anyone know what kind of candy bar this is? Spoiler alert, that's not fecal matter, that is a candy bar.
No big deal.
That's right, yeah. Baby Ruth. So another homework assignment, watch that movie if you get a chance. Excellent
Not
movie.
until you watch Osmosis.
But a kid drops a Baby Ruth into the water, and Bill Murray, of course, eats it in front of these people and horrifies them because they all think it's like fecal matter. But that is actually pretty dangerous. You know, first of all, you're sick. Oftentimes, we've had pretty well documented outbreaks associated with sick kids in public pools. And yes, there's chlorine in that water, but there's some chlorine-resistant organisms. So you get like diarrheal material into that water. And to your question, Aaron, like yes, that in a confined space like a pool, you can get a lot of people sick in that water if that kid is sick or the index patient. So yeah, there's nothing specific about it. It's just an indication if you have a high coliform count that you might have some contaminated water. Moving into a little bit of ANP, just so we can get our bearings on the GI tract. You guys have had GI yet? Starting tomorrow? Oh, cool. Okay. All right. So in terms of the colonization of the entire GI tract, we know that you're colonizing the mouth, right? You have a lot of stuff in your mouth and your esophagus. But what happens when we get down into the stomach, where the pH is around like 3? Can anything survive? Yes, some things can, but they have to be tolerant to acids. They have to be able to resist really low pH. They have to be tolerant to the urea, which most microorganisms aren't. So you have, it's like a desert in there, but you do have some species that can survive. You have some lactobacillus, you have some hardcore yeasts that can survive in the stomach, and you have a really important one called Helicobacter that we'll talk about probably tomorrow. Once we get into the small intestine, things change pretty quickly. You've got the duodenum right here, so the pH starts to already elevate and rise once you get into the duodenum. And then you make your way down the jejunum and the ileum. What's happening in terms of the pace of the food that's moving through your small intestine? Is it moving at a fast pace or a slow pace? Compared to the large intestine, I'll be more specific. It's pretty fast, yeah. So food is flowing through pretty quickly, and the bacteria in the food that you ingest are also flowing through quickly, which has implications for their ability to latch on and adhere to the intestinal epithelial cells. So this too, even though the pH is starting to level out a little bit and become physiologically neutral, you still have far fewer bacteria that can colonize the small intestine than the large intestine. Once we're talking about the large intestine, meaning colon, we have legit like bacterial paradise. This is where our bodies accept them, as welcoming them as we ever get to bacteria. So we have a lot of different variety, huge diversity of different genera that can be present in the colon, and tremendous numbers as well, 10 to 12 colony-forming units per mil. So huge amounts, and they are working for us. They're producing these short-chain fatty acids, SCFAs, that are important for our health, butyrate, propionate, acetate, vitamin synthesis, like vitamin D, vitamin K. So they do a lot of important things for us in terms of metabolism. Now moving back to the cell wall structure of Gramineatus, we've talked about Gramineatus cell wall structure before, but now we're talking about it in terms of how it's used for epidemiological purposes when we're talking about different strains. When we're strain typing enteric bacteria, we have 3 really good antigenic markers that we use on the cell surface. One, you remember the outer membrane of gram-negatives, it has that LPS, right, the lipopolysaccharide. And we've talked about this before, lipid A is on the interior of that, you remember lipid A. Then you have kind of a middle layer of polysaccharides, a mix of lots of different saccharides. And then on the outside is what we're getting at in this figure. That's the O polysaccharide. It is really good as this distinguishing marker for different types, different strains of bacteria. So we use that as an antigen by using commercial antibodies to see what strains these different bacteria belong to. The second major antigen are flagella themselves, so the bacteria have flagella, which most of these Enterobacteriaceae members will have flagella. So that's called an H antigen. And you do want to remember this is H antigen. So flagella, with an H on the end, maybe help you remember that. And then lastly, it's the capsule itself, capsule or K antigen. Sometimes it's called V when you're talking about salmonella isolates, but usually just K for capsule. So that also we use for epidemiological purposes. An example here is E. coli 157H7, this strain you keep hearing me talk about. It's the most important one. This isn't my area, but I know of a few other super pathogenic ones, like O116 has been associated with some big outbreaks, some other ones, but this one we will talk about today. So it's an important one, but what it means, if you've ever wondered what O157H7 means, is that it possesses that O polysaccharide group 157, and there are probably like 200 of these different antigens. And then for H7, that is the seventh flagellar antigen on that. So when we get outbreaks, if we can link all of those together, we can establish some sort of pattern and see if we have a common source, so we can track down the common source. All right, let's do a little bit of short-term recall, see how you do. Start with the LPS. What was the antigenic portion of the LPS? So we're looking at the box at the bottom here. O polysaccharide, yes, very good. What about this one at the top? It's pointing to this line, that red line. Flagella, also known as what antigen? H antigen, very good. And then this one that is kind of pointing to the outer surface of this bacterial cell is what? K for capsule. Okay, virulence factors. We can we go through this fairly quickly. Endotoxin we don't need to talk about anymore. I think you have a good sense of what it is and what it does. But remember, now that we're talking about gram-negatives, we're talking about endotoxin, and that is important for the virulence of these bacteria. Capsule we've talked about at length before. Capsule is really important for the Enterobacteriaceae as well. Phase variation, antigenic phase variation, we have not talked about a great deal. So what that means for an infection, if a bacteria wants to sustain a long-term infection, you need to be able to evade the immune system. So one method of doing that is simply to encode genes that express different antigens on the surface. So if you attract attention because your flagella has a certain motif, and that closest raise antibodies to that, simply induce expression of a different set of genes and start producing a different antigen, different type of H antigen. There are a couple other bacteria that do this outside of Enterobacteriaceae, so we'll talk about this theme again in the future. We've also got a secretion system, which we briefly talked about, but for the toxin-producing Enterobacteriaceae, they do need this secretion system, this mechanism of being able to get the toxin outside of the bacterial cell. So the type 3 secretion system, which is what is shown in this diagram here, is this very complicated, elaborate molecular machine that is on the outside, or it's embedded in the bacterial membrane, and allows those bacteria to produce those exotoxins and secrete them outside the cell. Sequestration of growth factors, that's primarily going to be iron. You can tolerate loss of a considerable amount of your own iron to bacteria every day. You have an RDA of however many milligrams of iron you need in your diet. You lose 10% of that to the bacteria that are residing in your gut, that's fine. But if you have a serious infection, you can become anemic over time. So, and that's if you have pathogenic bacteria in there that are heavily sequestering iron from you in your diet. And then antimicrobial resistance. A lot of these organisms that we're talking about, at least a couple of them, are escape pathogens. Some of them just have lots like multi-drug resistance, so that's considered another virulence factor. Enterobacter, E. coli, those are escape pathogens. So is Klebsiella. Yes, sir.
I just wanted to go back to the antigenic maturation now. So, is it fair to say that the regulated expression of the OKH antigens is like it's an inducible change?
Yeah, I think so, because there would probably be some sort of stimulus that, some environmental stimulus that you need to switch an antigen if you're capable of doing that, and then you switch the antigen. Otherwise, like, you do need to have these functional things, they serve functions, so you need to express something. But when it's time to vary that, that would be an example of inducible gene or operand. Okay, talking about E. coli, this is definitely the marquee organism for this genus, Escherichia. There are some other mildly pathogenic members in this genus, but Escherichia coli is by far the most clinically relevant one. We have, as I said, a lot of it in our gut. We mostly associate E. coli infections with intestinal infections. So diarrheal disease is the main thing. But because some of the virulence factors it carries are present, and because of the different opportunistic conditions like wounds or even like other conditions as well, you can get extra intestinal infections as well, urinary tract infections, meningitis, things like that. One of the main virulence factors are adherence factors, the ability to adhere onto these surfaces and establish infection. They also are usually going to be flagellated and mobile. So we can assess that in lab as well. We have different media types that can assess whether or not bacteria are motile. With these guys, at least on this image, you see this bacteria, this E. coli, it has a bunch of really long flagella extending out from the surface with shorter hair like fimbriae that would be more used for attachment. But the flagella, when they're extending in all directions like this is called a paratripous flagella pattern. It just means they're all over the surface of the bacteria. And it'll help them kind of swing around and navigate those viscous fluids in the body, in the gut. In terms of pathogenicity, most of these E. colis that are colonizing you are non-pathogenic. So in this cartoon, we're starting off with a non-pathogenic E. coli, just has its normal chromosome, but through horizontal gene transfer mechanisms that we've talked about before, it can acquire virulence traits by plasmids, by elements called transposons that we haven't talked about, but those are just mobile genetic elements that can jump from organism to organism. Phages are an important source of genetic transfer as well. So all of those can lend themselves to going from a non-pathogenic bacteria to a pathogenic bacteria. That can happen in the gut. So endogenously this can happen, or you can simply like eat an undercooked hamburger and get it exogenously. You get a virulent strain of E. coli that way. In terms of these different types of E. coli that can cause diarrhea, there are several important ones to know and to understand. These are referred to as pathotypes. And again, like they may not start out being pathogenic, but they can acquire those resistant or those virulence genes through different mechanisms. So these are just the pathogenic diarrheal types of E. coli. We'll also talk about the extra-intestinal infections as well. On this little flowchart that I've done, I've striated them according to whether or not they're non-invasive or invasive. We'll start off talking about the non-invasive ones, but when you study these, you want to kind of study them like this, whether or not they're non-invasive or invasive. So these non-invasive strains or pathotypes we'll talk about separately and individually. This E-tech is a toxin-producing E. coli, enterotoxigenic E. coli is what it stands for. It has two different toxins coming out of it, so these aren't subtypes of the organisms, these are just the toxins that it produces and elaborates. And then over on the invasive side, we have a toxigenic type of E. coli called Shiga toxin E. coli or S-tech. And then a subtype of that is EHEC or enterohemorrhagic E. coli. Okay. And then a non-toxigenic invasive type as well, enteroinvasive E. coli. So all those are spelled out over on in this little box on the left there. We'll talk first about enteropathogenic E. coli. So this is a non-invasive bacteria. Its main virulence factor is the fact that it can really tightly adhere to the surface of these intestinal epithelial cells. And it does it by the production of bundle-forming villi, or BFP. There's a BFPA gene, and when that is expressed, it forms these really, really tight adhesions with the host cell. This is also an interesting organism because it expresses its own receptor. It's called a transmessin intimin receptor, a TIR. It produces that, and then when this cell touches or binds to that enterocyte, that receptor gets embedded into that host cell, and it creates a channel for it to directly dump these BFPAs and establish like a really, really intensely strong connection between the bacteria and the host cell. So what happens is that triggers cytoskeletal rearrangements in the host cell, and that's what you're seeing in that artificially colorized electron micrograph on the bottom there, are cytoskeletal rearrangements in the host cell that produce these pedestals that the bacteria can sit on. In terms of the pathology that causes, it eventually causes the destruction of the microvilli on the surface of those enterocytes. So that's what's responsible for the diarrheal disease that you get when you have an infection causing EPEC, enteropathogenic E. coli. The target demographics that are mostly impacted are going to be kids, infants, and very young children. They tend to get this more often than adults. Because it's not invasive, you do not get perforation or penetration of those barrier cells. They stay intact, and you're not going to get bloody diarrhea like you would with some of the invasive types. A couple things to associate with EPEC, enteropathogenic E. coli: pedestal, patchy, platform, pediatrics. Okay, and importantly, we do not see toxins produced with this one. So it's pretty much, again, that pathology is because of the destruction of the surface through the attachment and effacement. The next pathotype is EAEC, enteroaggregative E. coli. With this bacteria, it too is staying outside of that enterocyte, so it is going to be important. It attaches to these things, to these target cells, but it doesn't actually penetrate into them either. This one does produce some toxins. It produces toxins like yeast and PET. I can't remember what those stand for, but basically what those toxins do is once you form a channel or pore into that enterocyte and it starts dumping those toxins in, those toxins will cause the dysregulation of those enterocyte cells, so they stop functioning correctly. Because of that, you get diarrheal symptoms. The demographics here are, it too can cause a lot of infant diarrhea, but it can affect all ages. So you see this a lot in traveling populations. Traveler's diarrhea, this is commonly associated with your pathotype with travelers. And we do get outbreaks in developed countries like the US, like Japan, like some European countries as well. Many of these are associated with like the developing world where they don't have high standards of hygiene and the water supply might be compromised. But stuff like EAC, we do see outbreaks here in the United States. This electron micrograph, you can kind of see the pathology a little bit. They form these stacked layers on top of the cells. ETEC, this is the last non-invasive pathotype that we'll talk about. With ETEC, the T you can remember for toxin, but also traveler's diarrhea too. This is another important one associated with traveler's diarrhea. We don't see a lot of ETEC in the United States, but as the name suggests, it does elaborate a couple of toxins. It elaborates a heat-stable toxin called ST and a heat-labile toxin called LT. Not all strains produce both of them, but they certainly can. Those are the more virulent strains. That heat-labile toxin behaves very much in the same manner as cholera toxin. They too have to be able to bind and adhere, so they produce a colonization factor to do that, to adhere to those enterocytes. But the toxins themselves are responsible for the dysfunction of those enterocytes and the diarrhea that it causes. In general, all of these different types, these pathotypes of E. coli can be tricky to discern different types. So with this one, the nice thing is we have a good PCR we can use to see if that gene is there, or the genes for ST and LT. That's very commonly done to diagnose. And we also have immunoassays where we use antibodies to see if they have the presence of LT or ST in stool. With IEC, we've got now an infection in the large intestine specifically. A couple of major O serotypes that are associated with this. EAIC, strains closely related to Shigella, they are essentially the same as Shigella. They behave the same way, they cause the same disease in the same populations. Really, the only difference between EAIC and Shigella is the fact that they're different bugs themselves. But otherwise, they present the same way. That might have implications if you're wondering, like, why would it matter for patient care? That might have implications for which antibiotics you treat them with, for the resistance patterns of those bacteria themselves, maybe even slightly the clinical picture as well. Like, this tends to be considered less severe than Shigella, but overall, thought of in very much the same way. What's different with this is now we're talking about a full-on invasion of those enterocytes. So we haven't seen that before, but these bacteria have ways of penetrating into those enterocytes and becoming intracellular. So they can escape destruction of phagolysosome, they can actually multiply and replicate in those cells. And then rather than, once they've multiplied and destroyed that cell entirely, which is what they do, and that's what's contributing to the really terrible symptoms associated with this, they can actually jut into the adjacent cells and continue to infect laterally like that. So they don't even have to expose themselves outside of the cell. Because of that, you get really horrific cases of diarrhea. Sometimes bloody diarrhea is associated with this. And that lends itself to the word dysentery. The word dysentery means bloody diarrhea. So this is bacillary dysentery. This slide, I think when I was reviewing this deck, it was looking to be a bit heavy, so it's broken up into the next two slides. You should have two slides that talk about STEC and rehab. So we'll start with this one. You guys all have this slightly more manageable learning, right? It's an important one, that's why there's so much material on it. For starters, this is one we talked about that can be colonizing cattle. So when we have outbreaks, we have big outbreaks. It gets into our cows, that gets into our meat, and when you hear about really terrible E. coli outbreaks when people are ingesting undercooked beef, this is oftentimes the culprit. So that isn't only really severe, but it's common as well. Last semester we had a big outbreak. I haven't been following ProMed as much as I normally do, so I don't know if there's been a recent outbreak, but it's not unusual. We get a couple big ones a year from this, where people, multi-state outbreaks, different fast food chains have been implicated. McDonald's had a big outbreak. Jack in the Box has had outbreaks. So definitely a big deal. Very low LD50. This too has the ability to destroy these cells and produce toxins, but it's not actually invading into the cells. So unlike EAIC, here where the bacteria are getting into and invading the cells, it's not doing that. It's destroying the microvilli, like we saw with EPEC, and it's also secreting some really gnarly toxins, Shigatoxin 1 and Shigatoxin 2, STX1 and STX2. Those toxins, you don't have to know what they do other than basically kill those cells, but they are encoded by lysogenic bacteriophages, which, as you know, can dump in those virulence genes and cause those strains to pick up those genes that encode those toxins. When you have an antibiotic treatment for a really serious case of diarrheal disease, remember that the rapid destruction of these bacteria, if they're gram-negatives like this, is associated with adverse effects, endotoxin release. Yes?
So, Shigatoxin causes actually forms that little pillars where we need that, that's why it's right.
Yeah, that
It's worse because you're actually causing necrosis.
Yeah. So the pathology is twofold. You have actual pathology caused by the same sort of attachment and effacement that we saw with other pathotypes. So you're destroying the function of those enterocytes, basically with that microvilli, is to increase surface area, aid in absorption, right? So you're no longer able to do that. But then in addition to that, you have toxins. Exactly, yeah, so those cells themselves are destroyed by the toxins. So the pathology is kind of like much, much worse when you're talking about EHEC and S type, because you have both of those virulence factors acting in coordination. Yep. In terms of the name, Shiga toxin producing an enterohemorrhagic enterohemorrhagic E. coli, EHEC and S type. For this course, you can treat them as the same, as synonymous. But of course, there is a difference. There is a subtle difference, and that is that all EHECs are S-Tex, and not all S-Tex are EHECs. If that makes sense. Shiga toxin producing defines the pathotype. They produce Shiga toxin. But not all of those strains result in hemorrhaging of the intestinal tissue, so not all of them are going to be EHECs. But just to keep it as clean as possible, people use them interchangeably, just know that there's a subtle distinction between those terms.
So these are trying to be invasive because of the endotoxins, but the cells, the bacteria cell extension cells. Is that right?
Well, because the cells themselves are destroyed completely by those toxins and by the attachment and effacement as well, that's why it's considered invasive, because it will destroy the barrier function of the cells. It's not just diarrhea, but the barrier function is destroyed. So you can get bloody diarrhea with enterohemorrhagic E. coli. Yeah. But is there invasion going on? Technically, no. You're just destroying the barrier toxins. Yeah. All right. So slide 2 of S-Tech, E-Tech is more about the clinical presentation, which we've already talked about a little bit. Depends on the strain. But if you are articulating or elaborating both STX1 and STX2 and you have a fair inoculum, you can get these really severe symptoms of bloody diarrhea. This can potentially lead to really serious things like dehydration, which can potentially be fatal. When you have loss of integrity in the intestinal barrier, you also open yourself up to secondary infections as well. So that can also result in fatal cases. Or they can be fairly mild as well. So these strains have been isolated from mild diarrhea cases as well. There's a really important complication that you need to know about. It's called hemolytic uremic syndrome, or HUS. It doesn't happen with all EHEC infections, but what happens is when you have the disruption of this barrier, some of these E. coli can get out, and they can start causing kidney damage. The toxin itself can also contribute to the destruction of the kidney cells. So with people who have HUS, who develop HUS, usually we're talking about children. Almost all of these cases are related to children who have had EHEC cases. Their kidneys start failing, and they get into some of these toxic situations, and they have thrombocytopenia as well, which means they have a clotting issue. Platelets aren't working properly. So it leads to could be potentially fatal condition for kids as well. It's also associated, as the kidneys start shutting down, urine production decreases as well. So that's one of the early signs that somebody has HUS. This is a quick write-up in ProMed from a little over a year ago. This was in Argentina. Cluster cases of HUS happened from kids who had EHEC. So there were 9 cases total, and if you extrapolate, probably 10 times that number of EHEC cases, so about 90 people, just extrapolating on that, probably had EHEC, 9 people had HUS. There were a couple of those patients who died. All of them were under the age of 7. That's very typical for fatal HUS infections. Okay, I've got 13:53. Do you guys want to take a quick break? I'm not going to go that much longer today. It's up to you.
Yeah.
Like 20 slides long?
No.
Three more slides. Three more slides and she's done. If she would have said that, we would just push through.
She says a few and then 10 slides.
That's true.
Even 3 slides takes her.
Non-invasive PTA.
Right.
It takes the same 14 seconds. I just need a more. I don't even know.
My ass shoot.
Like this right here. You know how she's got like all of it? She made a big one. She was crazy, right? I didn't do all the grand talking on this. By the time the lawsuit, I think it's short meeting of what's different between each one. Each one runs what's different. It sounds like it's similar, but I mean, and the big thing is like, the catalyst, positive, negative, let you know whether it's
Yeah, with the latter.
she's going to start doing catalyst, positive, negative. And it's going to tell you what's
Bro, this morning I woke up, like hit me. I don't know why. Like, I don't know who like punched me in my sleep. I woke up this. I was mad at the coffee maker. I was mad at my dog. I was just mad. I don't know why. I had no reason to be mad. Humid ass air walked out my door.
Go in your
Oh yeah, because I have AC. Some makes it. Some of them. Even like it shows like 100 apartments.
You can put that. This was a conversation. So
But it's classic. It's got personality.
clutches is
Yeah, I keep forgetting to do that. This weekend, and I just kept losing time. And now it's it's still on right now. I'm trying to do it tomorrow after we get off.
I did like 20 left.
It was race Tuesday, I wish.
So like that covers road, like yeah.
That's for them personality.
That kid collapsed in my life.
I'm reading.
I'm gonna be terrified if you were. The rest of them have like all the problems right now.
Yeah, they're more elite. Give me a pillow. Oh.
I know.
Start punching the air.
I don't want
But you know, like, I just feel like I make sure they're you know, you know. I just try to say,
this. I mean, I'm pretty slow. Like, walk by.
How much do you learn 1 mile?
I snoozed my alarm until 5:30 this morning. Yeah. My alarm went up at 5 and I was like, she's going to say, which one causes diarrhea? It's like,
Yeah, and the problem is like they all have like similar sounding names too.
Yeah.
So like you start saying it and it just is all going to sound like Latin. Just like,
I'm about to come up with names for them. Make them sound
come up with names. You have to. You're gonna play. Have you ever watched? You ever watched The Office? How how you don't remember names?
Yeah.
He just does it like in the most inappropriate ways or whatever. Just like remember people's names by like drawing like very absurd connections. You know, I wish I could even remember an example, but it would just they're usually racially charged too. Like ways of remembering. Yeah, Michael Scott.
The funniest part of that show is like watching the bloopers.
Oh yeah, like I love when they get to each other.
Yeah.
You know, like
Elongate.
oh, but the Pilates.
Taking karate lessons online.
Taking karate lessons online. Yeah. Just like even got Dwight on that one.
It's funny because Jim like turns around, he's like, okay, and then just starts laughing.
Yeah.
That shit was great. I rewatched The Office, the entire Office on deployment, and then I watched Game of Thrones. House of Dragons.
How was House of Dragons? I haven't watched
I haven't finished it yet. It's
it. Is
good.
it good?
You're a Star Wars guy?
And then TCA, I got there like 2 weeks before the block started. So I watched the whole thing. So I started as a kid. I'd never seen the end. It's really well done. But it's so
So you watched like Andor and all that?
yeah.
I'm not done with Andor. So I watched. So like you know how it came out where it wasn't actually in order.
Yeah.
So I watched it in order, and I was like, "That's so weird." Because like you watch it and it's like, I don't know the order of them.
Like
Yeah.
Like as they came out, it was like timeline was here, and then the next one would come out, it'd be timeline here,
Right,
and then
you
it'd be
get to
timeline.
meet Luke,
Yeah.
like
Yeah,
in
yeah,
the
yeah.
beginning, but really, then you're starting with like Anakin.
Yeah, and then you're like, oh shit, and then you watch it in
the
order,
whole series.
the whole
Yeah.
like all the movies, and then you watch them in order. So it's game changer.
Just a couple more slides and we'll get you out of here on this sweatbox classroom.
Oh, we're here till 5.
Oh,
Yeah, we're here till 5.
Joke's on us.
oh, that sucks. Okay, well, maybe I won't hurt you quite as much, but I would like to. Being honest. All right, a couple points that came up over break that I want to underscore here. One is in terms of methods of detection for this stuff. If you have a patient that's presenting with bloody diarrhea, before we move on to the extraintestinal infections, not all of this dysentery is going to be the same. So as you're waiting for culture results, assuming you have culture capabilities, and as you're waiting for the selective and differential media types to tell you exactly what type of bacteria you have, and then going on to do strain typing using those antigens that we talked about. While you're waiting for that, another valuable thing is to look at the patient's stool specimen itself under a microscope. You look for the presence of things like neutrophils, and you can see inflammation in there, like mucus can sometimes be shed. All of that is sort of taken in the clinical picture as evidence that you have destruction or damage to the colon. So you shouldn't, in a healthy person, if you look at their stool specimen, you shouldn't see a lot of inflammatory cells in there. That's highly suggestive that they have a bad bacterial infection. And also with the presence of blood, if you have one of those invasive pathotypes like EIC or EHEC, STEP, whatever, you may not see blood in the stool. You may or may not. If you don't, we have some really sensitive diagnostics. Maybe some of you have heard it before. They're called occult blood kits, and they look for minute amounts of blood in the stool that you wouldn't be able to see necessarily. Certainly not just a gross specimen looking at the stool. And you may not be able to even see that many red blood cells in the stool specimen either. But we do have sensitive occult blood tests for stool specimens as well. All right, extraintestinal infections. The really giant one that you want to remember for the rest of your life are the ability for E. coli to cause urinary tract infections, UTIs and E. coli. There's a name for these guys too. If you hear UPECs, UPECs are uropathogenic E. coli. There are a couple strains that are associated with this capability, but mostly you're talking about just normal gut E. coli's that contaminate the urinary tract, and that's how you establish these infections. They adhere to those urinary epithelial cells. I don't know of too many toxins that are produced. I'll have to look into that and see if there's toxin involvement, but just the attachment and effacement contributes to pathology and progressing to getting a UTI like this. The reason why you have to know this is because 75%, over 75% of UTIs are caused by this organism. We've only talked about one other bacteria that contributes heavily to UTIs. Do you guys remember what that other one was? Well, yeah, with Proteus, we talked about that today, I'm sorry. But it was a gram-positive cocci. Starts with an S. Staphylococcus. Yeah. So that Staphylococcus is a major contributor to UTIs, and it's definitely one you want to know and associate. It's the one, like, classically on CAS questions and exam questions, it's like a sexually active female who gets a UTI, something to consider, especially on a gram stain. If you see gram-positive cocci and not gram-negative rods, you're thinking Staph Staphylococcus versus E. coli. Okay. Symptoms, flank pain, so this would be probably a more serious UTI. Fever potentially, they're really bad. Dysuria, painful urination. Then we have another really serious and important E. coli extraintestinal infection called NMEX, neonatal meningitis E. coli strains. K1, this is a capsule produced, strain with a capsule that has a type K1 that is heavily associated with NMEX cases. How do you think newborns contract NMEX infections? Probably during delivery most of the time, yeah. We contract this during delivery. Oh, yeah, it says it right on the slide, doesn't it? But it is like a part of the mom's gut flora, and the baby will pick it up or can potentially pick it up during birth. And then it can cross the blood-brain barrier and cause neonatal meningitis. We'll talk about a couple other bacteria that are associated with neonatal meningitis as well throughout the course. We've already talked about one also. You guys remember what that one was? It's a strep. Strep pneumo?
Yeah.
All right, well, we developed a vaccine against it because we didn't want to have our kids get meningitis. We don't have a vaccine against NMEX. No, that's GBS, that's group B strep, and that also can contribute to neonatal meningitis. So yeah, we've talked about two then: group B strep, strep agalactiae, and strep pneumo as well. So this is our third, but no vaccine against it. So this remains a really big concern for newborns that develop meningitis. Definitely one you want to know. And we have just the fact that these two are oftentimes associated with healthcare settings, nosocomial infections, transmitted in hospitals, surgeries, patient to patient. A lot of times, if it's really bad, that patient's immune system is already compromised, you can get sepsis and toxic shock. It can cause pneumonia. This isn't the first thing that would come to mind if somebody had a respiratory infection, but it is possible that it causes pneumonia as well. And then wound infections, which can be really virulent as well. But the source, the origin, is always going to trace back to where it likes to colonize, which is primarily in the lab. Like I mentioned, when you have stool pathogens in general, it's incredibly important that you use the right combination of selective and differential media to get at the questions you need answered. You don't need to know if there's non-pathogenic E. coli in that stool, because the answer is always yes, right? You don't care about that stuff. You care about the stuff that is clinically relevant. So because of that, PCR and those immunoassays will allow you to actually detect either the genes for those toxins or the toxins themselves with antibodies are really important to get those questions answered. The plates themselves, the differential plates, will tell you things like whether or not, based on like fermentation patterns, for example, this McConkey plate will tell you whether or not you have like an EAIC or a different fermentation pattern from another E. coli. But that is possible, but if you want your question answered, is this producing toxin? Rely on one of those methods like PCR or immunoassays. For strain typing, especially if it looks like you might have a large outbreak situation, it's important to be able to have those serotyping reagents on hand so you can start linking cases together and report that to EPI authorities, public health authorities. With extraintestinal diseases, we're talking about urinary tract infections again, we're talking about wound infections, we're talking about meningitis. With those specimen types, when you see E. coli, that's always clinically relevant. That's not something you want to disregard, especially in CSF. Okay, that's all the farther I wanted to go today. So I know you guys have got a bit of a break, but any questions for me about E. coli? Yes, ma'am. hospital-associated infections, hospital-acquired infections. Yeah, yeah. And on that point, you guys have a good grasp on what the word nosocomial means, right? Synonymous with hospital-acquired. Nosocomial. Got it in hospital. Question over there? Did you have a question? Okay. All right, guys. So we will. Actually, we don't have a lecture tomorrow. We have our next lecture 9 o'clock on Thursday. So I'll see you guys then. Just let me know if you have any questions. Finish off this deck then. So thank you guys, and I apologize for the duplicate slides and stuff like that. But all right, have a good rest of your day.